Yo, welcome to NucleoGenesis. The NucleoGenesis Project is a temporal framework for eukaryotic nuclear blueprint archive and reconstruction. And if you don't know who I am, my name is Dr. James Utley. I am your resident codebreaker data scraper. I am the mad scientist behind this. And today, welcome to the reading of the abstract. So what I'm going to do is just read through this abstract here, and I'm going to go through each part afterwards in later videos. But yeah, let's get it up. Let's get it going. All right, so nucleogenesis, this is version 2.0, and it introduces a temporally decoupled biocomputational framework for eukaryotic nuclear blueprint reconstruction. The fundamental innovation lies in separating data acquisition from reconstruction. And this is reconstruction of high fidelity chromatin architecture assays. They are performed on fresh biological materials at the collection time and also impregnating a dried blood spot card, serving as the resilient long-term genomic and methylomic archives for future reanalysis and sequencing technologies that will happen in the future, right? This is a dual stream approach in addressing critical information from prior frameworks. The biological impossibility of recovering native chromatin conformation from desiccated substrates or dried out substrates, and it is formalized through an information theoretic decomposition. It distinguishes trying to define distinguishing features that are chemically persistent. That's your sequences, that's your methylation. Now, it also goes through through other layers, trying like the contacts, the accessibility, the histone state, the chromatin architecture, and that's all wrapping up the nuclear information. The pipeline comprises of 4 coupled modules with explicit data provenance tracking. That's the nucleosampler, that's your quality control, the nucleobuilder, which generates a tiered nuclear blueprint manifest, which is the most important part. It's a hierarchical, provenance-aware data structure that comprises of the gene, the methylome, the contacts, the expression, and the uncertainty layers that are mathematically distinguishable from the direct measurements. Now, it is statistically imputed features via reference-based contact, imputation, and cell type deconvolution. And that's the second tier. Model predicted architectures from sequence-based neural networks, that's the third tier, and explicit placeholders for future technologies are the fourth tier, so that's like the uncertainty layer. Now, nuclear reconstruction employs constraint weighted polymer physics simulations, which is incorporating a backbone connectivity, excluded volume, nuclear confinement, and A-B compartment segregation, where constant influence derives from tier-dependent weighted functions, and that produces a 3D model per locus position with an uncertainty that is quantified. It's a tiered stratified, and it provides tiered stratified contact satisfaction scores, and that is under the nucleo reconstruction module. Now, the last one is the nucleosynthesizer, and it orchestrates scalable computation through an architecture built on Rust, Python, and TypeScript, and it implements future-proofed archival via version-controlled nuclear blueprint manifest evolution. It's a deterministic content addresses hashing and the technology slots for anticipated assay modalities, including long read, dried blood spot sequencing, and partial methylomics. Like, so basically the fragments of the information that you receive as it matures in the future, you can be able to add it to the file. That's the nuclear blueprint manifest, or the NBM. Now, with the current present formal frameworks for data integration under heterogeneous confidence, let's go. So nucleogenesis delivers the most complete digital, or it aims to provide you with the most complete digital nucleo replica achievable from current technology. And that by doing that, it includes a phased genome sequence, the decade stable methylome profiles, and uncertainty that is quantified to try to build a 3D model of the chromatin architecture while establishing an infrastructure that's compatible with the future synthetic biology and advances in synthetic biology that could be used to reconstruct the nucleus. So that is the reading of the abstract. Of course, I paraphrased a bit, and this is a very dense paper, and I'm terrible at reading out loud, so don't judge me. But for the next video, we're going to go through all the contents of this paper. It's 39 pages, and here is all of the sections. And so we're going to go through each phase, which is the dual stream sample acquisition and management, which really makes this, in my mind, kind of special. So you have like an information build, as well as you have a fresh sample to manage. Also, you have the blueprint synthesis and all the future stuff, confidence weighted and spatial reconstruction, which is very cool and has everything weighted in terms of simulation and annealing and etc. And then a future, kind of opening up the window for future proofing orchestration. So then as technology grows, that you can update with basically version control. So it's like a digital version control of your nuclear blueprint manifest or NBM, and so that grows. So as the future matures, so does your manifest, if possible. Okay, and that is the NucleoGenesis Project for today. Thank you for joining me. Again, my name is Dr. James Utley, and I'll see you next time. Peace.